Well, I want to go ahead and start with a slide we all like to see, and this is, we've all heard that 65 is the new 45. And this is great, but there's just one little problem. When I'm 65, I want to beat this. Nothing can really get us to this. And so that's been my research. I'm a medical researcher focused on trying to find the cure for human aging. I've been in biotech for 31 years now, focusing the first 20 or 10 to 15, let's say, on cancer, heart disease, and inflammation research. And then in the last like 19 years, I have been totally focused on trying to figure out a way of turning this into this. And telomeres have become my favorite thing to work on right now, even though it's still a theory. Let me first get my name here, Bill Andrews. I'm the founder, president, and CEO of a company called Sierra Sciences. We're totally devoted towards trying to figure out how to cure human aging. But the problem, as I see it, is something called telomere shortening. This is a thing that seems to cause a lot of problems in human health, and what I'm trying to do is I'm trying to cure telomere shortening disease. I just want to fix this disease, cure this disease, because I know it plays a big role in human health. Our telomeres shorten. There's nothing we can do about it, and I'll tell you later, but it shortens and shortens and shortens. It's a scientific fact that it happens, and our chromosomes can't exist after it gets to a certain length. And so whatever we're going to do to cure aging, to get us to live hundreds of years, we're going to have to solve this problem. To explain what a telomere is, we need to zoom in on a human being. And we zoom in deeper and deeper into the cells, into the nuclei, and we get to the chromosomes. And the telomeres are actually found at the very tips of our chromosomes. Okay, now chromosomes are measured in units called bases. And what they are is not relevant for right now, but a typical chromosome is about 100 million bases in length. The telomere is only about 15,000 bases in length. At least it is when we're first conceived. So as soon as our cells start to divide, and inside the womb, we go through a lot of cell division, our telomeres shorten quite drastically. And so by the time we've gone through enough development to become a newborn baby, our telomeres have shortened from 15,000 bases down to 10,000 bases. The numbers are approximate, but it's a fact that this telomere shortening does occur. As we develop even further, and our cells progressively divide more and more and more, our telomeres get shorter and shorter. And then we get down to 5,000 bases, our cells lose the ability to function, and that's when we typically die of old age. So this is a process going on. Let me repeat this. We are conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. And telomere shortening, in my opinion, appears to be the clock of aging. There's a theoretical maximum that people have learned about a long time ago, that we can only live to be 125 years. Nobody's ever known why, but this clock, this telomere shortening clock, explains the 125-year theoretical maximum very well. And there's absolutely nothing we can do to stop this shortening. No matter how well you eat or exercise and do everything your doctor tells you to do, you can't stop this shortening. This is the only clock of aging that has ever been discovered. There are other things that do correlate with aging, but almost all of them have been correlated, have been shown to be turned off if we prevent the telomeres from shortening. So it seems like telomeres are the master clock that seems to regulate everything else. And again, this is a theory that I'm working on. So we need to figure out a way to prevent this telomere shortening. And I want to say that I can actually take blood from any of you. I can measure the length of your telomeres, and I can tell you how old you are. And more importantly, I can tell you how long it'll be before you die of old age, just by measuring the length of your telomeres. This pioneering work, this discovery was so significant that these colleagues that I worked with almost 20 years ago now, Elizabeth Blackburn, Carol Greider, and Jack Szostak, were awarded the 2009 Nobel Prize in Medicine just because of this research, for their pioneering research in this field. And I was awarded second place for United States Inventor of the Year for figuring out a way to lengthen telomeres. Scientists have now reversed aging in mice. Okay, scientists at Harvard, specifically Dr. Ron DePinto, who's now the head of MD Anderson, he actually, using this technology of telomere biology, was able to reverse aging in mice. He actually made old mice into young mice. And I'll be talking about that again a little bit later. But that's featured in this article, where I'm referred to as the man who would stop time. The best example that I know of that telomeres actually affect aging is that some people are actually born with short telomeres. They're also born with a mutation called lamin A, but it's been shown now that the lamin A mutation is the cause of their short telomeres. So these kids, they suffer from short telomere disease. Their telomeres are short, and then they stay short, and they have a life expectancy of about only 20 years. And they suffer from all the same age-related ailments that normal old people do. And if we could figure out a way to prevent the telomere shortening, this would actually be a cure for this disease. And actually, reversing telomere shortening could actually make them live totally normal lives. But right now, that's not the case, and there's only about 15 kids in the world at any one time with this horrible disease. But it's not the only thing. Scientists have been finding out that telomere length is correlated to practically every disease you've ever heard of. People, somebody gets sick, they measure the length of their telomeres, they find out that their telomeres are short. And this just shows some of them. On the left-hand side, I've got some of the diseases. On the right-hand side, I've got some other health-related areas that telomere shortening plays an important role in. And so I'm just going to talk about a few of these. First is cancer. It's been shown that cancer is actually affected by the length of your telomeres. When telomeres get short, I want you to think of like a chromosome, like a shoelace. Okay, you take your shoelace off your shoe, stretch it out. Well, the telomeres are the very tips of that shoelace. Well, when the caps of that shoelace get worn away, then the shoelace starts to fall apart. The same thing happens to your chromosomes. And when the telomeres get really short, then you start getting chromosome rearrangements and DNA mutations. And those rearrangements and mutations cause the different expression patterns of genes that lead to cancer. And so short telomeres plays a very big role in causing cancers. Also, one of our best defenses against cancer is our immune cells, our immune system. When the telomeres in our immune cells get short, then we lose the ability to fight cancer. And I'm going to be talking about that in a little more detail in another disease in a second. But if we could prevent telomeres from getting short, this would be a way of preventing cancer. And if we could keep telomeres long in our immune cells, this would be a good way to fight cancer if you already have cancer. And so keeping telomeres long is a really good thing. I have a common expression I usually say, and that's bad things happen when telomeres get short. We don't want short telomeres. We want to do everything possible to keep our telomeres from getting short. And I'm sorry to say there's very little you can do right now to do that. Stem cells. Now this isn't a disease, this is a whole field. Stem cells. Most people don't know this, but most doctors working with stem cells, with the exception of maybe the stem cells from our fat, they have to culture them outside the body to grow and make enough of them. Well, during all the cell division that's occurring, the telomeres get short. So if somebody took stem cells from your bone marrow, cultured it, and then injected it back into you, the stem cells are actually older than you are, even though they're your own cells. And that's because the telomeres have gotten shortened during the process. So I actually go to a fair number of stem cell conferences. If you do a Google search on me, you'll see I've published a lot on stem cells. And there's a lot of stem cell scientists looking to us at my company saying they need a way of preventing telomere shortening, especially for the older people. This is a really big problem. The older people, stem cell technology just doesn't work because the telomeres are too short, and they get shorter during the process. So this is another area where we are working on. Now, AIDS is a big problem. AIDS is actually, we consider our flagship project, because we're going to be testing AIDS patients in our clinical studies when we have something ready for clinical studies. And this is because when a person gets infected with the AIDS virus, HIV, the HIV causes accelerated telomere shortening in immune cells. You know, when a person gets infected with the HIV virus, nothing happens for years. But the telomeres in the immune cells get shortened at an accelerated rate to the point where the immune cells end up dying of old age because the telomeres got so short. Then the person doesn't have any immune cells. Then they get all these secondary infections, including cancer. Like I was mentioned before, telomeres cause cancer, short telomeres. And the best example you've heard of is Kaposi sarcoma. Kaposi sarcoma is caused by short telomeres in the immune cells, and the body loses the ability to fight cancers. Remember back in the early 1980s when doctors were first identifying patients in hospital beds that had AIDS, they were really surprised that they had no T cells, no immune cells. Well, we now know it's because of the accelerated telomere shortening. So what we want to do in our clinical studies is we want to show that we can allow AIDS patients to live normal lives even though they're still infected with the virus. The virus just keeps shortening the telomeres because telomerase inducers, things that lengthen the telomeres further, will keep them from, will keep their immune cells from dying off. Now, before we can do clinical studies on humans, we have to do preclinical studies on animals. And so the pet market is something that we're going to be really working on too. Cats actually have a disease very similar to AIDS caused by a virus called FIV, feline immunodeficiency virus. And so we are going to be testing cats to show that the cats can actually live normal lives too. But we actually expect a very fortunate side effect, and that is the cats are going to get younger. And that's not the goal of the clinical study, but that's what we expect to see. And what do you suppose is going to happen to people's interest in our research when they actually see these cats get younger? And what do you think the FDA is going to do to prevent us from actually getting this to people? So we don't know what's going to happen yet, but you know, medical tourism will probably play a role one way or another. My company, Sierra Sciences, founded in 1999 in Reno, Nevada, is focused on finding ways to lengthen telomeres. That's all we do, nothing else. We've been doing this for 14 years now. We've spent $33 million during those 14 years so far. During this process, we've identified the regulatory mechanisms that cause telomere shortening, and we have discovered chemicals and supplements that now disrupt some of these things, some of the mechanisms that are involved in telomere shortening. Why do telomeres shorten to begin with? And so to answer this, what I want to do is I want to use your imagination a little bit here and pretend that a chromosome is actually like the top layer of bricks on a brick wall. So let's get rid of the bricks, just leave the brick wall, and this is a chromosome. It's a long string of beads. Think of the bricks as the beads. And when a cell divides, the chromosome needs to be duplicated or replicated. And so inside the cell, and this is more accurate than you would believe what I'm about to show you, but inside the cell there's going to be a brick layer that goes along laying a new row of bricks, okay, to make the new chromosome that's going to be identical to the old chromosome, so it's essentially replicated. Now this is a long, arduous process. Remember, chromosomes are like 100 million bases in length, and so this has to be accurate because any mistakes will lead to mutations. Now where we're really interested though today is what's happening at the very end, so the telomere. Okay, so when the brick layer gets there, there's going to be a problem. You're going to see that the brick layer unfortunately is standing on top of the wall, so the brick layer can't replicate the very tip of the chromosome. Telomeres do not shorten by something chewing them away. It's not an active process. Well, they do a little bit, but the main cause of telomere shortening isn't something chewing them away. It's the lack of a process. It's the cells don't have the ability to replicate the very tip because that brick layer falls off. The brick layer inside of a cell is called DNA polymerase I. It gets there, it cannot replicate the very tip, so the new chromosome is shorter. So now, this cell divides, it gets ready to divide again, the brick layer comes along, laying more bricks, and again the brick layer is going to fall off. And as a result, the telomeres are shorter. And every time it gets shorter and shorter. Again, no matter what you do, no matter how well you eat, no matter how much you exercise, no matter how much you do, everything your doctor tells you to do, you can't stop this. I call this basal level telomere shortening. Now, there's other ways that telomeres shorten, because I said you can't slow it down or stop it, but you can sure accelerate it. And that's anything related to an unhealthy lifestyle. Smoking, obesity, lack of exercise, psychological stress, all of these things cause a generation of free radicals that will clip your DNA and cause accelerated telomere shortening. So you don't want to do those things. You want to lose weight, you want to quit smoking, because this is an active process. There's a lot of, and I call this accelerated telomere shortening. I mentioned before that there's been some significant studies that have come out on telomere shortening. There's 2 studies I'm going to show just the press releases for. This is one study done by Elizabeth Blackburn, the Nobel Prize winner, where her and her team looked at over 100,000 different people in a Kaiser Permanente study. They got blood samples from a long time ago that were stored, and then got recent blood samples, at least the most recent they could, and they found that people with the shortest telomeres died sooner. Okay, there's a very so people the same age, the ones that had shorter telomeres died sooner than the ones that had longer telomeres. So this is showing that telomere length is a good predictor of how soon it'll be before you die of old age. Another study, a lot more controlled, was done in the Seychelles Islands with birds. There's populations of birds that are they don't have a lot of predators, but they did a big study on measuring the telomere length of them, and they found that the shorter the telomeres, the sooner they died of all causes. Okay, so it's just, you know, it's not just aging, it's your ability, when your telomeres get short, you get a high degree of getting all the other diseases too that can kill you off. So we've got to keep our telomeres long. These 2 studies, that's what they're saying. Okay, so the big dilemma that faced a lot of scientists a long time ago is that if telomeres shorten every time a cell divides, why is it our children are born young? Why aren't they born old? Because reproduction requires cell division. And so wouldn't you think that their telomeres would be shorter than yours if you had to go through a lot of cell division to make a child? So this was a dilemma. People were wondering, there had to be something in our reproductive cells that prevented telomere shortening. And this is where I actually entered the picture. I led the research that discovered this enzyme called telomerase. This is a cartoon. The green is the DNA molecule, the very tip of the telomere. This factory-looking thing is this enzyme called telomerase. And what it's showing here is it's adding nucleotides or bases. Remember, it's in units of bases. These bases are being added onto the tip to lengthen the telomeres. This is what's happening in our reproductive cells. So let's go back to this bricklaying model. In our reproductive cells, the brick layer is still going to fall off the end of this wall when it gets there. But like an angel, telomerase comes in and replaces that missing brick. And so this is what happens in our reproductive cells, and this is why our telomeres don't shorten. Now, the next question is, what happens if we get telomerase in all of our cells? And this is research that I did shortly thereafter, after discovering telomerase. We put the gene for telomerase into normal human skin cells, first grown in a petri dish, and then further experiments. And we showed that when this is like, I want to say almost 20 years ago now, when we put telomerase into these cells and the telomere shortening stopped, these cells stopped showing any signs of aging. They essentially became immortal, growing in the petri dish. Since then, so we at the same time we did experiments to show that if we did the antisense of telomerase and gave it to cancer cells, we could accelerate their aging and cause them to die. So the company I was working with, so we could extend the lifespan, almost make human cells immortal by putting telomerase into them. But the antisense would actually, instead of increasing lifespan, decrease lifespan, and was a good way to accelerate the aging of cancer cells and getting them to die very quickly. One thing that happened just as I was leaving is that we took these cells that were from, we took cells that were essentially from a 90-year-old person, treated them with telomerase, grew them into skin on the back of a mouse, and showed that these cells were young by every biomarker of aging that we could ever imagine. The controls, where the cells weren't treated with telomerase, they looked like old cells. They were wrinkled, they blistered easily, lots of different tests. Gene expression analysis was done. When these telomerized, we call it telomerized, when these telomerized cells were grown on the back of mice, they were young by every biomarker of aging we could imagine. All right, so since then, in fact, this is just like 2 years ago now, Dr. Ron DePinto, I said he's the head of MD Anderson now, he was at Harvard, he actually engineered an experiment where he could turn the telomerase gene off and on in mice. He allowed the telomeres to get really short in the mice. They looked like really old mice. They could barely walk around, they couldn't reproduce, their brains didn't work very well, they couldn't remember how to go through a maze, their organs were failing. They then fed them this drug that they had synthesized that turned the telomerase gene back on, and before anybody gets excited, this drug only works on these mice, okay? So it doesn't work on humans and it won't even work on other mice. But when they fed these mice this drug that turned this telomerase back on, they got young. And this is a news report from Diane Sawyer.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous!
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
Well, I don't believe we have as long to go as she was referring to. I think it's going to actually occur sooner. But I think this is really exciting. Telomere shortening and related to aging is still a theory. But this is one of the best, let's say, not proof of concepts, but sure support of concept that I've ever seen. Dr. Maria Blasco in Spain repeated the study using a different technology. In this case, she used gene therapy to infect mice with a gene with the viruses carried a telomerase gene into the mice. What she found when she did this, she only did one treatment, whereas the other ones, they had long treatments of telomerase turning on. She just did one short treatment and saw a 24% extension of lifespan in these mice. So I'll tell you, this is really getting pretty exciting. Now, that's pretty much what everybody else is doing. Here's what we're doing. at my company. We're searching for a pharmaceutical that's going to turn on the telomerase gene. We want to turn us into those mice that got younger. I want to look like those two people. Remember those first two slides I showed? That's what I want to be when I'm 65. That's what we're doing. We're searching for a pharmaceutical that's going to stop or reverse telomere shortening in humans. And so to describe how we're doing it, this is showing a gene. The gray thing is part of the chromosome. Telomerase is produced by the telomerase gene inside of cells. Next to so this is at least in our reproductive cells, you can see this blue thing, the gene is producing telomerase. Next to every gene, there's a regulatory element. It's kind of like a light switch that turns genes off and on. So in our normal cells, we have a protein that binds to this regulatory element and shuts the gene off. This protein is called a repressor, and that's what occurs in every cell of our body except our reproductive cells. So for some reason, all of our cells are turned off. And so what we're doing is we're searching for drugs, chemicals, supplements, anything that is going to bind to this repressor and dislodge it and just turning it on. And one thing I want to point out is that this is like a dimmer switch. We can find some chemicals that work at very low levels to barely turn telomerase on, and examples of that are product B and TA65. These things are just barely giving us detectable telomerase expression. But what we want to do is we want to really produce some really high-level ones, and we have produced a lot of high-level ones, not at the supplement level, but at the synthetic chemicals. We've now discovered synthetic chemicals using robots like this. This is a picture showing two of our $1 million robots that will be testing like up to 4,000 different chemicals a day, looking for anything that turns on telomerase in human cells. And of course, we also have scientists working all the time on all this kind of stuff. We have now found 39 families of chemicals. We found like 900 different chemicals. We've been able to put them into 39 different families of chemicals that will actually turn on telomerase. I mean, I'm talking about hundreds of times better than TA65 and product B. And we are 3 years away from testing this in humans. So the planet is about to change, in my opinion. Better get used to it. We're all going to be looking forward to this, I hope. And thank you very much.